Humans have synthesized tens of thousands of synthetic industrial compounds in the last century. These compounds have been used for everything from making better plastics to killing pests that eat our crops. From DDT to thalidomide to bisphenol A to perfluorocarbons, we've learned time and time again that these chemicals can have unintended consequences for our health and for the health of the planet. And the lasting legacy of this chemistry has been the unexpected persistence of many of these chemicals, both in the environment and in our bodies. In fact, from the bottom of the sea to the poles of the earth, you can find man-made chemicals in the air, in water, and in the bodies of most living things. In fact, if we measure the blood of any person alive today, we find hundreds of these synthetic compounds present, many of them acquired simply through the food we eat or the personal products we use. And in many cases, we know that these chemicals have adverse effects on our health and on the health of future generations, who inherit the consequences of our exposure to chemicals. With industrial chemical being such an indispensable part of modern life, how then do we fix this problem? And is there some way to design chemicals that won't be so persistent, or to predict if chemicals will be persistent before we ever use them? Historically, the way we used to do this is to predict persistence from the structure of chemicals themselves. We know that if chemicals are likely to dissolve in fat, they're also likely to cross cell membranes and accumulate in our bodies. Make chemicals that are less fat soluble, and you can expect that they will not be very persistent. But this doesn't solve the problem completely, because many of the chemicals that are the most persistent don't behave as we would expect from knowing their structures. So where else can we look for solutions? One way to approach the problem might be to focus on biology. What if we could ask a cell what chemicals it can deal with and which ones it can't? It turns out that the flood of genomic and cellular information in the last decade lets us do just that. We, like all living things, have evolved strategies to deal with chemicals we encounter in the natural world. Those chemicals are present in the food we eat and the air and water we breathe, and through many millions of years of evolution, our bodies have been tuned to recognize and eliminate those natural structures we encounter most frequently. Within our bodies, this action happens inside cells, each cell having a membrane that keeps the insides in and the outsides out. You might think of cells as being a bit like nightclubs, with people waiting to get in, others dancing around, and yet others coordinating the show and making sure it all runs smoothly. From the nucleus to the membrane, cells are exquisitely complex machines with thousands of parts, each with a specific function to play. And all of these parts work to accomplish two basic functions. One is to help the cell grow and divide, and the other is to help the cell interact with its environment and adapt to changes. The gatekeeper of the nightclub is the cell membrane, which at once helps a cell communicate, brings in nutrients, and keeps out toxicants. In fact, this membrane has an exquisitely tuned capacity to reject or accept toxic chemicals. The way they do this is by using a family of proteins called multidrug efflux transporters. The transporters are like bouncers hanging out at the membrane and keeping harmful chemicals out. Meanwhile, the inside of a cell is like a virtual fortress of defenses designed to protect our DNA against damage. And behind the scenes of the nightclub, defenses are on the lookout for toxic chemicals that escape these defenses and need to be broken down or locked away where they can do no harm. By now you should be asking, if a cell has all of these defenses to keep itself going and keep natural chemicals out, why do they all fail to keep environmental chemicals out? It turns out there's several reasons. One reason is that synthetic chemicals are so good at doing their job that they just slip past the defenses without being recognized. Another reason is that we get exposed to chemicals at stages of life that are not adapted to deal with them, and the chemicals confuse our endocrine systems. The last reason is that we sometimes just overwhelm the defenses. The bouncers can keep out 1 or 2 rowdy characters, but when 100 show up, turns out we can use this knowledge of defenses to make safer chemicals. In fact, we already do something very similar, except instead of making sure chemicals are kept out, we make sure they get past the defenses. For example, when we test a new pharmaceutical drug, we measure its level of interaction with multi-drug transporters or drug metabolizing enzymes to make sure it'll actually slip past the defenses and get into our circulation. If a drug is eliminated by the transporter or metabolized by the enzyme, we can predict it won't be effective at getting around our body. So what if we turn this idea on its head? That is, test industrial chemicals in the same way we test pharmaceuticals, but this time look for chemicals to be recognized by the cell's bouncers and easily eliminated. And at the same time, we could flag the chemicals that slip past the bouncers and sort through the thousands of chemical structures, identifying those that are likely to be persistent versus those that might be problematic. You might even tune chemical structures so that the bouncers can easily spot them and get rid of them. Put another way, we make all the industrial chemicals look like the kinds of characters that never should get into a nightclub and never start trouble in the first place.